Hello, I'm Jessica Jamsi Cantamayor, reporter of Group 3 for the VMA Vet Parasitology 412. So today, I will be discussing about fecal culture and modified Baermann technique. The material used in the study are the following: grams of fresh feces from goats, glass jar, sterile vermiculate, gloves, tongue depressor, 1% solution of solution carbonate, compound microscope, water, petri dish, pipette, and Logos iodine. Measure 10 grams of feces. Moisten the feces by putting small amount of water and mix it thoroughly. After that, place the feces in the bottom of the glass jar provided. Then add sterile vermiculite or sterile rice hulls and mix to give approximately 50-50 mixture. Close the lid of the jar and incubate at room temperature for a week. Larvae will migrate up to the side of the jar and may be seen as white mold-like material. Add water to the culture jar to the brim. Invert a petri dish over the mouth of the jar, then turn the jar so that it stands in the petri dish. Pour about 10 to 15 ml of water into the dish and allow the jar and dish to stand overnight. Collect the larvae by pipette the following day from the petri dish. Examine the live larvae, then add a drop of Logos iodine, which kills and stains the larvae. Hemoncus contortus, also known as the barber's pole worm, is a very common parasite and one of the most pathogenic nematodes of ruminants. Adult worms attach to a vomesal mucosa and feed on the blood. This parasite is responsible for anemia, edema, and death of infected sheep and goats, mainly during summer in warm, humid climates. Hemoncus are the parasites of the abomasum of ruminants. It has a buccal cavity armed with a lancet. Its length is up to 30 mm. The male has a symmetric dorsal ray in its bursa and short, wedge-shaped specules. The white, egg-filled uterus of the female spirals around the blood-filled cut, giving rising to the so-called barber pole appearance. Adult female Strongyloides westeri are located deep in the mucosa of the small intestine. The larvated eggs pass in the feces. In the environment, a rhabditiform larvae develops within each egg, which then hatches. The rhabditiform larvae from each egg can either molt twice, developing into infective third stage or filariform larvae, which is the homogonic cycle, or molt 4 times and develop into free-living adult males and females. These free-living adults can then produce rhabditiform larvae which molt twice, developing into the filariform larvae, which the heterogonic cycle. Goals to controlling Hemoncus contortus includes preventing heavy exposure in susceptible hosts, reducing overall level of pasture contamination, minimizing the effects of parasite burdens, and encouraging the development of immunity in animals. The treatment used in Hemoncus contortus are fenbendazole, thiabendazole, ivermectin, and albendazole.